So next in the module 2, we will be looking more into the various parameters in FECB equation, and something associated with that, something important associated with that is called the temperature response function. So temperature is affecting all the biochemical reactions that are happening in plants, including the biochemical reactions happening during photosynthesis. So in order to predict the biochemical reactions which are a function of temperature, we use this temperature response functions. Some key temperature response functions and how they are applied in this model BioCro and various other models that are simulating these photosynthetic processes like Rubisco limited photosynthesis, RUBP regeneration limited photosynthesis, and triose phosphate utilization limited photosynthesis is what we'll look into right now. So one such important equation is temperature, or an equation is the Arrhenius equation. It is a formula for the temperature dependence of reaction rates proposed by Svante Arrhenius in 1889. So why is this equation important? It is because the model temperature dependence of enzymatic reactions in photosynthesis and respiration is given or is represented as using the Arrhenius equation. So we had learned about Vc max, that is the maximum carboxylation rate, and J max, which is the maximum electron transport rate, and also TPU, which is the maximum triose phosphate utilization rate. All these parameters in photosynthesis are associated to temperature and is represented using the Arrhenius equation. So Arrhenius equation is given by this formula where K is equal to A e to the power minus EA by R into T. So K is a rate constant, A is a frequency factor, EA is the activation energy of the enzyme, R is the ideal gas constant, and T is the absolute temperature in Kelvin. So this is how this is the Arrhenius equation. Now, when it comes, this is purely in terms of how it is represented in the model. So, in order to avoid this A term, because it is often very difficult to determine this A term directly, we do a process called normalization, where where in here what happens, the A is redefined in terms of a known rate constant at a reference temperature T ref. Here we are taking the T reference as 25 degrees Celsius or 298 Kelvin. So what we do is we know the temperature for this equation where K25 is the rate constant at the 25 degrees Celsius temperature. A is the frequency factor, EA is the activation energy, R ideal gas constant, and 298 is the temperature in Kelvin. So for 25 degrees Celsius, this is how we can write the Arrhenius equation. Now when we rearrange this Arrhenius equation to find representation for A, the equation becomes like this, where A is equal to K25. This whole term comes into the denominator, and when it comes into the numerator, this minus sign will go. So this will be the final equation for A. So K, rate constant at 25 degrees Celsius into the exponential term with the temperature term represented in Kelvin as 298. So we know this term, and we also know that the rate constant is at 25 degrees Celsius. Now when we substitute this value of A in the main equation of Arrhenius, we will get the rate constant is equal to K25 e to the power EA by R into 298 into again this term that is there in the Arrhenius equation. Now when we again do some rearrangement, we will get it in this form. This part of the equation, which is EA by R into 298, is represented by a scaling parameter which is dimensionless. So the final equation becomes like this, where the rate constant for an enzymatic reaction is the product of the rate constant at a referent temperature, where the referent temperature that we are considering here is 25. So we are putting it as K25 into exponential of C minus EA by RT, where C is equal to the activation energy by ideal gas constant into temperature at the reference temperature, which is 25 degrees Celsius in Kelvin is 298. So this is how the temperature response parameter or temperature response function is represented in Biochrome and in various other models also. So where is this Arrhenius equation used in our model Biochrome and how is this Arrhenius equation connected to the FECB equations parameters? Now, if you see here, we had talked about something called the Michaelis-Menten constant, Kc. So, Kc is also a temperature dependent function and is represented by e to the power Cmin. It is the same representation except that this K at temperature 25 degrees Celsius, Kc is equal to 1. So, we get that K is equal to, here this K is what is Kc is equal to K25 is 1 for the Michaelis-Menten constant Kc, and exponential of C is equal to 38.05. The scaling parameter is equal to 38.05. EA activation energy is equal to 79.43 joules per mole, and here temperature, any temperature. So this equation is to calculate the Michaelis-Menten constant for any temperature, and here in place of K25 it will be equal to 1 because as part of normalization, that is what we assume that this Kc should be 1. I mean K at 25 should be 1. So here this is the scaling parameter, this is the activation energy, and R divided by R into whatever R into the temperature term, and for any temperature we can use this equation to find out what is the value of the Michaelis-Menten constant. So same applies for the Michaelis-Menten constant for oxygenation. Here the scaling parameter is 20.3 and activation energy is 36.38. When it comes to the condensation point, which is another term in the Rubisco carboxylation limited photosynthesis, the scaling parameter is 19.02 and activation energy is 37.83. So in this equation, we were able to find out Kc, K0 and tau star or condensation point. Now Vc max at any temperature is equal to the Vc max at 25 degrees Celsius. That is, this value is equal to the value of Vc max at 25 degrees Celsius into the exponential of scaling parameter, which is 26.35 in this case, and activation energy is 65.33 in this case. So all these values we have obtained from these two papers by Bernacchi. They have conducted several experimental, they have done various experiments and a lot of optimization to obtain these values. So most of the C3 simulation of C3 plants, generally this is the scaling factor and activation energy values used for the corresponding terms, and the representation is through Arrhenius equations representation. So all of these terms are temperature dependent, which means that for this equation at a particular temperature, if the value is A1, then at a different temperature the value becomes A2 because of the variation of temperature. So this is an indication of how internal carbon dioxide as well as temperature plays an important role when it comes to photosynthesis. Now, I kept on saying about Michaelis-Menten constants. So, Michaelis-Menten constants is based on this Michaelis-Menten kinetics, where a key parameter, this Michaelis-Menten constant is a key parameter in enzyme kinetics, which is representing the substrate concentration. So, here if the substrate concentration in here is carbon dioxide and oxygen concentration. So, here Kc is representing the substrate concentration, which is nothing but carbon dioxide concentration, at which the enzyme will operate at half its maximum reaction rate. So, Kc is the concentration of intercellular carbon dioxide at which the enzyme Rubisco will act at half its maximum reaction rate. Now, here reaction rate is Vc max for carbon dioxide, that is the maximum rate of carboxylation. So Kc is actually the concentration of intercellular carbon dioxide at which Rubisco will the Rubisco's action yeah wait it is the carbon dioxide concentration when Vc max is equal to half of its maximum value. So if Vc max is equal to 100, the concentration of carbon dioxide inside the leaf at which Vc max is equal to 50 is giving us the Michaelis-Menten constant. So that is how this term, that is a significance or the relation of this term to this equation. In the same way, K naught also is the concentration, substrate concentration means concentration of oxygen when the rate at which oxygenation will take place at half of its rate. That is, if oxygenation rate is say 100, what is the concentration of oxygen when the rate of oxygenation is equal to 50 is giving us this Michaelis-Menten constant for oxygenation for O2. Now, in this is what this is a repetition of what I had just said. In FECB model, this Michaelis-Menten constant is what is the representation of Rubisco, Rubisco's affinity for carbon dioxide. So we know that Rubisco has an affinity for oxygen and carbon dioxide. So at what concentration of carbon dioxide is Rubisco Rubisco has a tendency to help with the carboxylation reaction, or what is its affinity for carbon dioxide to conduct the carboxylation reaction is what the Michaelis-Menten constant gives. Now, how is this Michaelis-Menten constant estimated? It is estimated to this temperature response function that we had just discussed, where this is the scaling factor, this is the activation energy, and at any temperature value we can estimate the value of Michaelis-Menten constant and then put it in the FECB's equation to estimate the photosynthetic rate. Now, a higher Kc means lower affinity of Rubisco for carbon dioxide, requiring higher intercellular carbon dioxide for efficient carboxylation. So, if the value of Kc is high, it means that the Rubisco has got a lower affinity for carbon dioxide. And if the value of Kc is low, it means that Rubisco has a high affinity for carbon dioxide. So it is working efficiently in whatever carbon dioxide concentration is present within the leaf. But in here, we see that a higher Kc value means the Rubisco's affinity for carbon dioxide is less, as a result of which it can the reaction of oxygenation is more probable to occur over there, as a result of which photorespiration will occur. Now, same in the case of oxygen, the Michaelis-Menten constant is representing the affinity of Rubisco for oxygen and oxygen concentration at which photorespiration will occur at half its maximum rate. That is the reaction happening here is oxygenation, and what is the substrate concentration, oxygen concentration when the photorespiration is going to occur at half its maximum rate is giving us the value of the Michaelis-Menten constant for oxygen. So it is also a temperature response function and is given by this equation similar to the Kc value, except that the scaling parameter and the activation energy varies. And again, since Rubisco is an enzyme that is binding both carbon dioxide and oxygen, this ratio of K naught by Kc, which is the Michaelis-Menten constant K naught for oxygen and Michaelis-Menten constant for carbon dioxide, this ratio has an important role in determining which reaction will happen, whether it will whether photorespiration will happen or whether carboxylation will happen. So a higher K naught means less competition from oxygen. That is, there is higher chances for carboxylation to occur. So the photorespiration will be lower and carbon dioxide fixation will be better. Lower K naught means Rubisco has a higher affinity for oxygen. So photorespiration will photorespiration is the process that is going to compete with carboxylation and more probable to happen. So this is how we can read or we can obtain the Michaelis-Menten constant and analyze the results of photosynthesis based on them. Now, in the next limited photosynthetic rate, which is RUVP regeneration limited photosynthetic rate, the equation to estimate this photosynthetic rate is J into Ci minus tau star by EC into Ci plus 2 into E naught into compensation point. So, in the earlier equations, we found out how to find this compensation point, which is equal to, which is also related to a temperature response function. So in this way, we can estimate the carbon dioxide compensation point. So these three values, tau star is something that we know. Now, another important quantity here is the rate of electron transport. So, unlike Vc max, it is J is just the rate of electron transport, and J is given by this equation Q2 plus J max minus under root Q2 plus J max the whole square minus 4 beta theta psi 2 into Q2 into J max divided by 2 into theta psi. So, in order to estimate this parameter in this equation, this is the formula used for it, where Jmax is the maximum rate of electron transport at a particular temperature. So, as Vc max is the maximum rate of carboxylation, here in RUVP regeneration limited, the maximum rate of electron transport is given by Jmax. Q2 is the maximum fraction of incident ppft that can be used in electron transport. So I had told that when light energy is absorbed, the light energy absorbed or light energy that is used is represented in the form of photons, that is micromoles of photons per meter square per second. So that is what we call as photosynthetic photon flux density. So Q2 is representing what is the maximum amount of ppft that is used for the electron transport rate. Now theta psi here is a linear light response factor, that again I will show the graph from which you can understand more better. And Q2 is given by the product of incident ppft, that is whatever light is available to the plant, not whatever light is absorbed by the plant, but whatever light is incident for the plant into one parameter called psi ps2, which is the maximum dark adapted quantum yield of photosystem 2. Now for understanding all of these parameters, we will have to go more and more deeper into the theoretical part. Here, since our main focus is modeling, I would just like to introduce these terms, because when you use Biocro, all of these equations are coming within the code. This is how so many parameters are estimated. So I just want to make everyone familiar with these, what these parameters are. So maximum dark adapted quantum yield of photosystem 2. So photosystem 2 is the chlorophyll center in the leaf, in the chloroplast, in the thylakoid membrane, where light energy first is utilized. So it is in photosystem 2 that the light energy is used for exciting the electrons, that is produced as a result of oxidation of water. So maximum dark adapted quantum yield of photosystem 2 is what theta ps22. So dark adapted quantum yield is, there are experiments where we close the leaf, keep the leaf and acclimatize it to a dark condition, and then open it and measure the rate of photosynthesis. So this practice of adjusting the leaf to a dark condition and then seeing what is the amount of light absorbed is called dark adapted measurement. So during that, what is this value of theta ps2 that we obtain is what is used in the estimation of this maximum amount of incident ppft that is absorbed by the leaf. Similarly, beta ps2 is also another parameter, another value of fraction of the absorbed light that is reaching photosystem 2. So when you multiply these two values into the incident PPFT and the light reflectance or absorbance, we get this value of Q2. So Q2 is used in this equation. Jmax, I said, is the maximum rate of electron transport. Theta is also the linear light response factor, and Q2 is estimated using this formula. So where does temperature response come? Temperature response play a role in this equation is over here. So here Q2 we know is given by this one. Now this, how do we obtain this 5 PS2? So 5 PS2 we can obtain it by measurement, but when we estimate it as a function of temperature. So this 5 PS2 as well as theta PS2 is given by the equation 0.352 plus 0.022 T minus 0.0034 TL square. So if you see this equation, unlike the previous equation that was represented by the Arrhenius equation, this looks like a polynomial equation with a plus bx plus cx square. So so the temperature response function over here is not Arrhenius equation. I'll come to that in the next slide. So in order to give an understanding about the values in this RUBP regeneration rate, we did look into two terms in the denominator. One giving us the electrons used for carboxylation and one giving us the electrons used for oxygenation. So when you come to the equation, there is a value of 2 you see over here, 2 into electrons for oxygenation. So why is this 2 in the denominator? Is because 2 cycles of photorespiration is required to generate one carbon dioxide molecule. So if there has to be one carbon dioxide molecule generated during the photorespiration reaction, there needs to 2 cycles of photorespiration needs to occur. So 4.5 electrons per carboxylations are required to regenerate RUBP via NADPH and ATP. So you can go back to the Calvin cycle to see that RUBP regeneration is making use of NADPH and ATP. So 4.5 electrons are required to regenerate one RUBP using this NADPH and ATP. Similarly, 5.25 electrons are required for every oxygen molecule for photorespiration to occur. So that is why when we come to this reaction, we give a 2 into 5. This value of E naught is used as 5.25 and EC is used as 4.5 in the model. Now I said there is this the value of phi is the theta is the convexity term for electron transport rate or linear response factor, which means it is this value. So when you see that the curve is changing from linearly increasing line to a steady line, this part of the curve is what it gives, or this part of the curve is given by this theta ps2 value mathematically, if you see. So this curve determines this convexity term or convexity factor or linear response factor. Now if in order to find out the phi ps2 phi ps2 obtained is 5 PS2 from the graph. If you need to obtain, you have to find the slope of this linear line. So if you consider this line and if you find the slope of that line, that value is what the 5 PS2 will be equal to. So from the graph, this is how we can obtain the 5 PS2 value. Now say we don't have the 5 PS2 value and we need to estimate the value. We can use this equation and what is the temperature at that point of the leaf to estimate the 5 PS2 value. So where is Arrhenius equation used in this RUBP regeneration limited? This equation is used to represent two important parameters, which is Jmax. Jmax is a function of temperature and is represented by the Arrhenius equation. Here the scaling factor Jmax is the maximum electron transport rate at 25 degrees Celsius. C is the scaling factor and 43.54 is the activation energy. Now we also learned about dark respiration. Dark respiration in the equation is also represented by this Arrhenius equation, where Rd is the respiration rate at 25 degrees Celsius. C scaling factor 46.39 is the activation energy. So if you use all these equations, we will get all these parameters that is required to estimate the photosynthetic rate. Now I had also shown that this 5ps2 is estimated using a polynomial equation. So this is how a polynomial equation is often represented. When we use the variable as the temperature, the polynomial equation is going to be a plus or c naught plus c1 into tl plus c2 into tl square, where c naught is the baseline rate, c1 is the first order coefficient, that is what is the first order differentiation per change in degree Celsius. This is the second order coefficient. So polynomial is going to look, it is going to be a curved line. So C1 will be the linear rate of change with temperature. What is the change in this term with with change in temperature, first order coefficient. If C1 is positive, the response is going to increase, and C1 is negative, the response is going to decrease. C2 is the curvature term, that is the second order differentiation of second order differential term. It is controlling the curvature of the response and accounting for the non-linear temperature effects, which is also very important. So C1 is giving us a more linear approach, whereas C2, the second order differential term, is giving us the non-linear temperature effects. If C2 is negative, the response is going to follow a parabolic shape, and if C2 is positive, it is going to curve upwards like this. So if C1 is positive, it is going to be a line, a straight line, and negative, it is going to be a straight line going downwards, declining slope. And if C1 is C2 is positive, it is going to be a curved line uh, upwards, increasing curved line. And if C2 is negative, it is going to be a curved line which is going below in the negative axis. So in photosynthesis, this both terms theta and phi, which I had shown in the graph, is represented at particular points. But if we don't have a graph to obtain these values, the values can be estimated if we know the temperature at which all these measurements are done using this equation, where theta is the baseline linear response. 0.018 is the first order coefficient and minus 0.0037 is the second order coefficient. So if you see here minus, this is what if you develop the equation for various kinds of temperature, this is how the curve will look like. Same in the case of 5ps2 also, this is a negative term, so this is how the curve will look like if you generate that equation for various values of temperatures. If you estimate the 5ps2 for say 25 to 45 degrees Celsius, the curve is going to be a curved line if the C2 is negative. Now there is another equation that is also a modification of the Arrhenius equation called John Eyring-Williams model. In this also there is a representation of this various terms in the FECB equation in the with respect to temperature. Here if you see, T is the temperature at which the measurement is made or where the function is being estimated, C scaling parameter, HA is the activation energy. So the numerator is the same, but in the denominator we get a denominator for the Arrhenius equation, where the denominator is 1 plus e to the power s by r. S is nothing but it is an entropy term influencing the deactivation energy. So apart from the activation energy, a deactivation energy is also considered, and it is represented in this form in the denominator of the Arrhenius equation. So this is the John Eyring-Williams model, or this is how the Arrhenius equation was modified to include the deactivation term. So where is this used? This is used in the triose phosphate utilization limited photosynthetic rate. We know that VTPU is the maximum rate at which triose phosphate is utilized, and it is given by this equation where VTPU is the TPU rate at 25 degrees Celsius, and here 25.5 is the scaling factor, 62.99 is the activation energy. If you see here, this equation 0.58 into TL minus 182, this whole term, I think R has been taken out and this whole term has been rearranged to get it in this form. So we don't have to learn all these equations. All of these are included in the Biocro model, and all the terms that are estimated. So when we give a temperature input, this is how all the terms in the FECB equations are estimated. So this is about the temperature response functions.